Get out of it, man. Can somebody state whether they hear me or not?
We hear you.
Okay. Okay, before we begin, are there any questions? Get rid of that. Should probably open another page here too. All right, I'm not hearing any questions, so that's good. I need to go in and get this changed. I'm not sure how to do that. Let me share my screen. All right, you should be seeing my screen now. Today is January 23rd, and we are supposed to be doing Lab 4 stains. Any question about any of that? Go to the lab. All right, Lab 4 stains. Simple stains, differential stains, and special stains. You do need to read a little bit in the textbook. Read about the size and the shape and the arrangement of bacteria cells from page 73 to 75. Read about the bacteria cell wall on pages 80 to 83. Read about the simple stain on page 65. Read about a differential stains, the gram stain from page 65 to 66, and the acid fast stain from page 66 to 67. And then read pages 67 to 68 about special stains. We'll talk about three special stains: the negative staining for capsules, the endospore stain, and then the flagella stain. And they are listing the pages to read here, mostly page 67 to 75 to 77, page 68, and then page 92 to 94, and then page 68 to page 77 to 78. There are some supplemental materials in this lab. There are video clips and a virtual lab. Your learning objectives are: one, be able to prepare a bacterial smear for staining. A bacteria smear is something you do to put really bacteria on the slide before you do any staining. So the bacteria smear is separate and independent from the stain or the staining. And students often get that incorrect. Two, understand the principles and procedures used when performing a simple stain, a differential stain, and special staining techniques. Three, be able to interpret the results for a simple stain, a differential stain, and a special stain. And then four, be able to define the following terms: a simple stain, a differential stain, a special stain. What's a coccus? What's a bacillus? What's a spirillum? What is peptidoglycan? What are gram-positive cells? What are gram-negative cells? What's the acid-fast stain? What is a primary stain? What is a mordant? What is a decolorizer? What is the counter stain? What is a capsule? What is an endospore? And what is a flagella? Any questions about any of those objectives? All right, bacteria are almost transparent. Therefore, it is necessary to stain them before they can be observed under the microscope. Now, it is possible to see bacteria without them being stained, but I think we covered this in the lecture. It's much easier to see them if they are stained. When preparing a bacterial smear for staining, you First, add a drop of sterile water on the clean slide, and then you add a small amount of bacteria in the drop of water. You mix the bacteria in the drop of water and then smear that over the surface of the slide, allowing the slide to air dry. The organisms are then heat fixed on the slide by gentle heating. And the purpose of the air drying and the heat fixing is to attach the cells to the slide so they're not washed off during the staining process that will follow. Any question about any of that? All right. A simple stain is simply using one dye to stain a bacterial cell. And you notice I've crossed out the word basic because it doesn't matter what dye it is. If you use one dye, it is a simple stain. It will stain all the cells the same, but you can tell different shapes and cellular growth arrangements of the cells. We'll talk about shape and the cellular arrangement in a little bit. A differential staining technique uses two or more dyes to provide additional information about the cell. The Gram stain is a common differential stain. And that's one of the two we'll talk about, the one we'll talk about the most. Lastly, there are special stains, and they're used to dye a particular bacterial structure, such as the cell capsule, an endospore, or the cell's flagella. Any questions about any of that? All right, here we're looking at cocci, and you can see the growth arrangement. First, not shown is an individual cocci, and that we call a singlet. And then there are diplococci, where there are two cocci stuck together. You can have four cocci stuck together, and we call that a tetrad. You can have lots of cocci stuck together in a clump, and that we call a staphylococci. And then the cocci can also be in a row, and that we call streptococci. Any question about any of that? There's also um, eight cocci stuck together, and I'm not seeing that here. I imagine it's shown later on in this lab. You also have bacilli, and there's bacilli shown there. You can have spirilla, and these are cells shaped like an S. Those are those there. You can have cells which are pleomorphic, and they take on many different shapes. This is sort of a cross between a bacilli and a spirilla, and that one's even not even bacilli shape. And then you can have coccobacilli. That is a cross between a bacilli and a cocci. Coccobacilli. Any questions about some common cellular morphologies and then the three basic shapes of the bacteria? The three basic shapes are the cocci, the bacilli, and then the spirilla. All right, let's talk about a differential stain. In 1884, you don't need to know the name, or the year, sorry, but you do need to know the name. Dr. Graham, Hans Christian Graham, devised the Graham stain. And you should capitalize the G in Graham because it's named after Dr. Graham. Any question about any of that? He made this stain to be able to differentiate between a patient's cells and the cells of the bacteria that were causing the infection. However, microbiologists used the Gram stain and they realized that it distinguished between two very types of bacteria depending on the bacteria's ability to retain certain dyes. The bacteria that stain and retain the crystal violet from the gram stain are classified as gram-positive cells, and they will appear purple from the crystal violet. While there are other bacteria that do not retain the crystal violet in the gram stain, and they are referred as gram-negative and appear pink, and they're stained by the safranin stain. Any question about any of that? And there's some cells stained with the gram stain. Let me blow that up. There's a gram-positive bacilli. There's a gram-positive or some gram-positive cocci. There is a gram-negative bacilli. There's a gram-negative spirillum. I don't think there's gram-negative cocci in this preparation. Gram reactions are based upon the bacteria's ability to retain the crystal violet during the gram stain. The cell wall composition of the gram-positive bacteria is structurally different than the gram-negative cell wall. Gram-positive microorganisms contain a thick peptidoglycan layer, while gram-negative bacteria have a thinner peptidoglycan layer. And that peptidoglycan layer is determining the characteristics of the gram stain, meaning whether the crystal violet will be stuck in the cell wall or it will be lost by the cell wall. Here you see a gram-positive bacteria cell wall, and it does have a plasma membrane. And then there's a thick peptidoglycan layer, which you can actually see some of the layers there. And the gram-positive cell walls are something like 30 layers of peptidoglycan up to hundreds of layers of peptidoglycan. Whereas here's the gram-negative cell wall, and you'll notice it's different. It does have the plasma membrane inside the cell wall. That's not part of the cell wall, by the way. And it does have a thinner peptidoglycan layer, between about 3 layers of peptidoglycan, one stacked on top of the other, to 20 layers. And this one, you can actually see some of the layers in there. There might be 6 to 12 different layers in there. And then it has this outer layer, which is similar to the plasma membrane. We call it the outer membrane of the cell wall. And it does have a phospholipid in it, which is the main molecule of the cell membrane. It does have some other molecules in it, and then it has this carbohydrate attached to it. We'll show that a little bit better in a future picture. Any question about that? This is the main difference, the peptidoglycan layer and the thickness of the peptidoglycan layer in the gram-positive cell wall and the thinness of the peptidoglycan layer in the gram-negative cell wall. All right. After bacteria have been stained with crystal violet, the Gram's iodine is used as a mordant in the Gram stain. A mordant is something that combines with the primary stain, and that's the crystal violet, to form the crystal violet iodine complex. This complex is larger than iodine alone or crystal violet alone, and so the complex is more likely to get stuck in the thick layer of the peptidoglycan layer, but will wash out of the thinner layer of the gram-negative cell wall. How that is washed off is you use the alcohol acetone solution as a decolorizer. It's applied only briefly to remove the crystal violet, and it will remove it from the gram-negative cells, but not from the gram-positive cells. The timing of applying the decolorizer is critical because if you apply it for too long, it will pull out the crystal violet from the gram-positive thicker cell wall. But if you don't apply it long enough, it won't pull out all of the crystal violet from the gram-negative cell walls. So the timing of the decolorizing step is critical, and it's the most important part of the gram stain. After you pull out the crystal violet from the gram-negative cells, the bacteria are colorless, and that's why we apply safranin as a counter stain to color the colorless cells, and they will pick up the color pink. Any question about any of that? All right, let me mention that some bacteria, like the mycobacterium, are not stained very well with the gram stain. The mycobacterium have mycolic acid in their cell wall, and mycolic acid does not allow water-soluble substances into the cell, and the gram stain are water-soluble. The mycobacterium are gram-positive, just they don't stain well. But if you use the gram stain appropriately, meaning you have young cells, they do stain gram-positively, but they're lighter than a normal gram-positive cell. Any question about that? And this is warning you that sometimes the mycobacteria, as well as some other bacteria, can appear gram variable. This is one species of cell, and the student or somebody who did it got the cells to stain both gram positively and gram negatively. Let me blow this up a little bit. What do you notice between the gram negative cells and the gram positive cell? Like this cell here is a very good gram negative cell, excuse me, gram positive cell, and this one there. And then that one there is a good example of a gram negative cell, and there's another good example of a gram negative cell. And right there, right here is another good example of a gram negative cell. What do you notice between these three gram negative cells and then these 1, 2, 3 gram positive cells? What do you notice? Nobody's going to take a guess?
The color.
Shape.
Well, yeah, the color. What do you mean by the shape?
Um, the the gram positives looks like they're caucus shape, right?
Yeah.
And then the gram negatives, so that's the pink, is the bacillus shape.
No, the gram purple is gram positive. It's bacilli.
Yeah. So I said opposite.
Well, that is one cell there. That is one cell there. That is one cell here. It's in between these two cells. So these are all bacilli. There's no cocci in this picture. Why did you think that this was a cocci? Let me ask you that.
It kind of looks like it has like little circles connected.
Yeah, what's happening is this cell is ghostly. You're seeing it like it's a ghost. The cell isn't entirely all there. And then that one there, and this one there, that's a little too blurry. It's better that way. And then that one there, these cells look broken. That one looks like a ghost. These are kind of ghostly, but they're broken. And the reason why they're broken is because they're actually dead cells. So that's why it looks ghostly or broken. This is a dead cell, that's a dead cell, and that's a dead cell. These, the cell wall, as well as the cell membrane, looks intact, and that is why they don't look ghostly. You can see a clear distinction between outside the cell and inside the cell. Whereas this and that one, it's not so clear where the cell ends and where the cell begins. And that's because they're dead cells. So my point is this preparation is an old preparation, and these gram-negative cells are probably dead cells. And then the gram-positive cells are the viable living cells. And that's why this preparation is staining both gram-negative and gram-positive, because some of the cells are dead and some of the cells are alive. And most of the time, actually all of the times I've ever stained the mycobacterium, whenever I had a student do the gram stain and it looked like this, I asked them, how old are these cells? And everyone always told me that the cells were at least 2 weeks old or older. And then I told them, on the gram stain, it only works correctly if you stain the cells that are 2 days old or younger. And so they were doing a gram stain on an old preparation of cells, and the gram stain does not work well on old cells. You should only apply the gram stain on freshly grown cells. So if the cells are over 2 days old, you should regrow them and then apply the gram stain. Any question about any of that? All right. So even with the mycobacterium, every time I've stained them, they've all appeared gram positive. Now, I've only stained maybe 2 species of the mycobacterium, so I don't have a lot of experience, but I have stained the mycobacterium a large number of times. I only got gram-positive cells. They will stain lighter, meaning they do not readily take up the stain, but they do stain gram-positively as long as you have the cells 2 days old or younger. So some tips to prevent the gram variable stain. Avoid using old cells. They should only be 2 days old or younger. Prepare a thin smear. If you have a real thick smear, it'll be difficult getting the stain all the way down to the bottom cells. And then if it's really thick, it's also difficult to remove the crystal violet from the lower layers with the decolorizing step. Heat fix the bacteria on the slide only after the smear is air dried. If you heat fix before the cells have air dried, the water will boil, and that will cause holes in the cell wall of the cell, and that will prevent the gram stain from working correctly. And then avoid overheating the cells. If you heat the cells, it makes the cell wall disrupted, and it looks like the cells have actually melted. Use fresh staining reagents, and then do not over decolorize or under decolorize the slide. Any question about that? All right. The other differential stain we're going to briefly talk about is the acid-fast stain. Some bacteria, namely the cells in the Mycobacteria family or the family Nocardia, have a waxy substance called mycolic acid in their cell walls. Particularly the Mycobacterium, they have a lot of mycolic acid in their cell wall. And these bacteria are able to resist acid alcohol decolorization, meaning they retain the acid fast stain. And we call these bacteria acid fast bacteria positive, meaning they're positive for acid fast stain. Any question about that? And then other bacteria, most of the bacteria in the world, are acid fast negative bacteria. There are two acid fast stains, the Zeal-Nielsen and the Kenyon. They both use carbol fusion as the primary stain. Um, they use a different mordant. Zeal Nielsen uses heat to drive the carbonyl fusion inside the acid-fast positive cells, while the Kenyan stain uses a high concentration of the stain to act like a chemical mordant, causing the stain to get inside the acid-fast cells. And initially, all cells will take up the carbonyl fusion, which is cherry red in color, and then the cells are decolorized using acid alcohol to decolorize the bacteria that are acid fast negative, but it will not decolorize the acid fast positive ones. You then stain the cells with either methylene blue or brilliant green and possibly crystal violet. I've never seen that. Usually it's methylene blue or brilliant green. It's applied as a counter stain. The counter stain is just a different stain than the primary stain, and it's a different color. And the counter stain will colorize the decolorized cells. At the end of the staining process, the acid-fast cells, meaning the acid-fast positive cells, will stain red, and the non-acid-fast cells, meaning the acid-fast negative cells, will stain either blue or green, depending on which counter stain was used. So if you look at acid-fast stain under the microscope, there will be red staining cells, which are acid-fast positive bacteria, and then either green or bluish cells, which are the acid-fast negative cells. Any question about the acid-fast stain? Remember, the acid-fast stain and the gram stain are the only two differential stains we're ever going to talk about. All right, lastly, let's talk about the special stains. The special stains stain a special structure within the cell. We have a negative stain, which is one of the special stains. So let me start this again. The special stains are a whole category of stains that can stain different things. The negative stain is a stain that we use to stain the glycocalyx. If the glycocalyx is firmly attached to the cell wall, it is referred to as the capsule. And so the negative stain is also called the capsule stain because it will stain the capsule. There are other glycocalyxes. We're not going to talk about them in this lab, but actually the capsule stain will not stain the other glycocalyxes. Any question about any of that? So if the glycocalyx is a capsule, the negative stain will stain the capsule. And usually it stains the cell lightly. And then it does not stain. Now, wait a minute, I didn't say that right. Usually the negative stain stains the background of the slide. It slightly stains the cell usually, but it does not stain the cell capsule. And because the background is stained and the cell is stained, the capsule does appear in your picture of the stain. The production of the capsule can be associated with the organism's virulence or its pathogenicity, as it helps protect the cell from being phagocytized. And so usually if a patient has a bacterial infection that has a cell capsule, it's usually virulent because the host will have a hard time removing these cells. And that's one reason why Streptococcus pyogenes causes strep throat. And then the patient oftentimes does not get better because the patient has a hard time removing the bacteria, attacking the bacteria, because the Streptococcus pyogenes has a cell capsule, which protects it. Any question about any of that? Another thing about cells that have a capsule is when you look at the colonies of these cells on a petri dish, the colonies look mucoid, and it's because of all the capsule surrounding the cells that makes the colonies look mucoid. Capsules are not easily observed with conventional staining methods. Like when you're doing the gram stain, it will wash away the cell capsule. So you cannot see the cell capsule in the gram stain. And therefore, we use a negative stain, which is a very gentle procedure to demonstrate the presence of the capsule. India ink and nigrosin are two dyes commonly used in the negative stain. Here is India ink, which lightly stains the cell. It's kind of their kind of grayish color, and then it darkly stains the background of the cell. And you note the clear region around the cell, that is where the capsule is. Here is a different capsule stain using two dyes. One dye stains the cell, another dye stains the background of the slide, and neither dye stains the capsule. So once again, the capsule is very visible in this preparation. Any question about any of that? By the way, it's called the negative stain because, like in this picture here, it looks similar to a photographic negative, which, if you remember, the Polaroid cameras used to have. Um, and that is the background is staining opposite of it looks in the under the microscope. Normally, the background is crystal clear because all it is glass and maybe water, and then the capsule is staining, and that's why it's called the negative stain. Any question about any of that? All right, an endospore stain is a special stain developed to stain the endospores. Endospores are metabolically inactive, hardy structures that are produced by some bacteria as a defensive strategy when the environment becomes unfavorable to the parent cell, and then the parent cells will make the endospore. The endospores can survive, but then the parent cell will die. It's just because the environment has become unfavorable. Typically, endospores are made by gram-positive bacilli, but not all positive gram-positive bacilli can make endospores. And mostly it's two families of bacteria that make endospores, Bacillus and Clostridium. So those two families. There is at least one other family, but it only has one member in the family, and I don't remember the name of the family. When the environment then becomes favorable, the endospores will germinate and they will return to their normal metabolically active vegetative state. So the endospore will germinate and become a bacilli. And then that bacilli will become metabolically active and can reproduce. The endospore itself is essentially not metabolically active. It's very, very low metabolism. And it's just a cell being held in stasis. It also has a different shape. We'll take a look at that in a minute. Oh, there I state the two genera that have the endospores are mostly genera Bacillus and Clostridium. Here are some clinically significant pathogens that produce endospores. Bacillus anthracis, Bacillus cereus. Anthracis causes the disease anthrax. Bacillus cereus can cause food poisoning. And then there's also Clostridium botulinum, which causes food poisoning, botulism specifically. Clostridium tetani, which causes tetanus. Clostridium perfringens, which causes gas gangrene and food poisoning, and Clostridium difficile, which causes antibiotic-associated colitis or pseudomembranous colitis when the patient gets diarrhea. And because this has an endospore, if you treat them one time with antibiotics, frequently the patient will get Clostridium difficile, survive in the antibiotics, and the diarrhea will come back on them. Any question about any of that? All right. The Schaefer-Fulton stain is an endospore stain which uses malachite green as the primary stain. Heat is applied to the mordant to help the primary stain get inside the endospore. The cells are then decolorized simply with water. The water will remove the malachite green from the vegetative or the perianthal cells, but it will not remove the water, will not remove the malachite green from the endospores. Safranin is then applied as a counterstain to stain the cells that are negative for the malachite green, meaning the decolorized cells. So the vegetative perianthal cells will appear pink from the safranin. The endospores will appear dark green from the malachite green. Let me see if I can blow this up. There we have an endospore which has come out of the cell. And there we have an endospore which is being made inside this bacillus right there. And there's another one there. And the green part is the endospore. And then the parent cell is the pinkish part of the cell. Any question about any of that? It's called an endospore because the spore is made inside the perianthal cell. All right, the flagella stain is the stain that stains the flagella. The flagella are long thread-like appendages, sort of like a whip that's attached to the cell's surface, and it gives the cell motility or locomotion, meaning if the cell can swim or move, it has flagella. And I guess I should clarify that a little clearer. If the cell is a bacteria cell and it can move, then that cell has to have flagella. A eukaryote cell can move if it has flagella, but it can move if it has pseudopods or if it has cilia. So an endospore cell can move by three different means, but bacteria only have one way that they can swim or they can move, and that is the only thing they have is flagella. Any question about any of that? So if the cell has flagella, it can move. Flagella are composed of flagellin. That's a class of protein. You can also count the number and the distribution of flagella on the bacteria surface. It is useful in classifying the bacteria. You need to know this for the lab, but I'm not going to quiz you on the flagella arrangement. You can have one flagella on one end of the cell, and that's called monotrichous. You can have multiple flagella on one end of the cell, and that's called lophotrichous. You can have multiple flagella on both ends of the cell, and that's called amphitrichous. And then you can have flagella all over the cell, and that is called peritrichous. Flagella are too thin to be seen using standard staining techniques, so special stains are required to view them under the microscope. And many of the special stains, what they do is you apply it more than once, and it puts on layers on the flagella, so layer upon layer, And then when you view the cell under the microscope, you can actually then see the flagella because it has so many layers coating the flagella. And here we see the flagella stain. What do you notice about some of these cells in here? Like, what about that cell? Does it look different than that cell in the number of flagella? Specifically, this one here. Is that different from that one? And maybe different from, well, maybe that one. How about that one? Is it different, that cell different from that one? It's a yes or no question, people.
Yes.
Hmm. There are a different number of flagella. These are all the same species of cell, but this one is peritrichous. Some of them are amphitrichous. Some of them are lophotrichous. And then the last cell I showed you, excuse me, um, some of them are monotrichous. And then the last cell I showed you had no flagella. Let's go back here. This cell there has no flagella. So what's going on if these are the same species? What's going on is when the cell is first made, meaning the cell just divides and then the cell is first made, it doesn't have a flagella yet because the cell hasn't made any. And then it will make one and it will be monotrichous. And then it'll make more and it'll be lophotrichous. And then it'll make more and it'll be amphitrichous. And then it'll make more and it will be peritrichous. So you count the number of flagella on a species and then you take the highest count. So this species would be called peritrichous,
So it's like the life stage of the cell.
the last stage of the cell. All the earlier stages, the cell just hasn't made enough flagella yet, and that's why it's appearing in different stages. Now, some cells will never become peritrichous, and they will only become amphitrichous. But the amphitrichous will also have the lophotrichous, the monotrichous, and then the no flagella. And then some cells will only go as high as lophotrichous. So you have to count the cell that has the most flagella and then give the arrangement from the highest count. Any question about any of that? All right. Excuse me. Sneezing again.
Bless you.
Thank you. Not sure what's going on. I did about 2 days ago, but I didn't notice it yesterday. But of course, I wasn't really paying attention. All right, know the terms of this lab. Don't need to know the references. I once had a student report that there was a, what do you call that? They clicked on the reference and it took them to a bad website where there was a, what do you call that? A bug? No, not a bug.
Virus.
Virus. Yeah, there was a virus on the reference. So probably best not to, but that was an exception. We did remove that link. All right, the lab for exercises. You're not going to be physically performing any of these activities, but you must understand how to prepare bacterial smears for staining. And remember, a bacterial smear is something you do before you do the staining, and it's independent of the stain. You must understand the principles and the procedures used for various staining techniques and be able to interpret the results using a microscope. So be able to answer the following. In procedure one, we're going to be preparing a smear for staining. So you can view the video clips on preparing a bacterial smear, and then another video clip on heat fixing that smear. And so that's the bacterial smear, something you do before you do the stain. You then can review the steps of making a stain. And here we have, excuse me, this is making a smear. First, you mix a small amount of the specimen in a drop of water, usually use sterile water. Spread it around on the slide, let it air dry. After it's air dried, you heat fix. That you do by putting it up to the Bacto incinerator, and that will cause the cells to stick to the glass of the slide. So they're not washed out by later procedures, such as the staining procedures. Let the slide cool, and now the smear is ready to be stained. And this is your first question, which I guess I shouldn't put it here. I should go to the worksheet. Now, I thought I removed the questions from your part, but I might have it still in mine. So the first question is procedure 1, question number 3. What are the 4 common errors when preparing a bacterial smear for staining? So list those there. Procedure 2, simple stain. View the following clips on a simple stain. Review the procedure of a simple stain. You simply add a dye. It doesn't have to be a basic dye. Usually it's methylene blue or crystal violet. You apply that dye to a bacterial smear. After about 1 minute, you gently wash off the dye with water. Shake off the excess water. Blot it, the slide dry. Let me show you how you do that. So you pretend this is your slide. You get a piece of bibulous paper. It's really thick, almost like cardboard. You rip it out, and then you fold it over so the paper is over the slide, and then you just press the paper, bibulous paper, down onto the slide. You do not take the slide and rub it against the paper or rub the paper against the slide, because that will remove cells. And then enough of the water will be removed that you can even use oil on the slide and then view the cells under the microscope. Remember, when you're starting and using the microscope, you should always start with low power and only use the fine adjustment knob after you've got the specimen in focus with low power, and then move the specimen to the center of your field of view, and then go up to the next higher power objective, and then repeat that procedure. All right, so here we have E. coli cells stained with a simple stain and Staphylococcus aureus cells stained with a simple stain. Let me warn you that this preparation has a lot of cells stuck together, and this is one cell there, that's 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 one cell there. Here, this is actually 3 cells, one there, and then a third one there. And once again, that's at least 2 cells, maybe 3. 1, 2, 3, actually 4 cells. So there's a lot of cells stuck together here. I will warn you, E. coli is not a spirillum. Okay. And then those are the Staphylococcus there. You can see them better. And it asks you, what's the cellular morphology, the shape of the E. coli? Do not give me the cellular growth arrangement, just the shape of the cells. What's the shape of the Staphylococcus aureus? And then for procedure 2B, give me the total magnification that was used to observe these cells. Procedure 3, the gram stain. View the following clip on the gram stain. Practice performing the gram stain in the virtual lab. Click on the virtual lab. It'll probably take a while for that to load. For one thing, I got to open up a new window. Oop, what happened there? There it goes. And there's the virtual lab. Read through the gram stains.
Hi, lab assistant. Well,
Read through it.
all yogurt.
Just gotta move stuff to it.
This is what. Now take a look at this yogurt. This yogurt. Let's. In the gram staining. Bacteria. We are going to. All right. Let's prepare our slides for gram staining. To prevent bacteria from contaminating our work area, we have wiped down the table with a 70% ethanol solution. We will also light a Bunsen burner to sterilize the inoculating loops we'll use later and help maintain a sterile working environment. You're going to use a striker to light the Bunsen burner. The gas is turned on low already, so bring the striker to the Bunsen burner with the flint side facing down and squeeze the striker to create sparks.
So just take your mouse and follow the arrow. The arrow is telling you what to do. And that brings the striker down to...
We need to dilute the yogurt before we put it on the microscope slide.
To get this procedure to work. And I'm going
to thin out the yogurt. That helps us see the... Start by placing 1 milliliter of the diluent into...
Turn that off. I'm just going to move to the gram stain. Anyways, it's telling you what to do. Oops. Come on. Sorry. Get this out of here. Material smear. It looks like they didn't allow it to air dry, but we'll just continue. You should air dry before you do that, by the way. All right, here's the gram stain.
We apply them in a specific order and rinse the slide off under slow running water in between each chemical.
So to do the gram stain...
The first is crystal violet. This is our purple stain, which will indicate gram-positive bacteria. Using the dropper, squeeze out enough drops to cover your sample. Let that sit for 60 seconds. Now rinse it under slow running water. Now use the bottle of Gram's iodine.
Okay.
Gram's iodine will fix the dye to the cell walls of the bacteria and make it permanent.
This is how you go through the gram stain. Please go through this because it'll actually take you through all the steps of the gram stain. Any question about the virtual lab? So you do need to know how to do the virtual lab, so please go through the virtual lab. Many you do need to know the gram stain, and the virtual lab will go through it through the procedure with you. Here we're going through the gram stain. Oh, where's my mouse? Oh, shoot. There it is. So first, you apply the crystal violet. It will cause the cells to stain purple, and all the cells, both the gram-positive and the gram-negative, will look purple after applying the first stain, the crystal violet. You then will wash that off with cold water, and then get the worst of the water off. Then apply the Gram's iodine. Iodine is a mordant. It will combine with the crystal violet to make a crystal violet iodine complex, which is larger than the crystal violet alone. When you apply the crystal violet, the cells will take on a more purple color before they were a blue purple color, and it's not showing you a change in the color, but there it is. And all the cells will then be a purple color, whereas before they were a blue purple color. You then apply the decolorizer, and you have to be very careful about it so that you decolorize only long enough to pull the crystal violet iodine complex out of the gram-negative cells, but short enough so you don't pull it out of the gram-positive cells. And if you don't do it long enough, you won't remove all the crystal violet iodine out of the gram-negative. And if you do it too long, you will remove it from the gram-positive cells. Anyways, the alcohol acetone is usually used as the decolorizer, although there are other decolorizers. In the video of the gram stain, I think they only use alcohol, in which case you apply the decolorizer for a longer period of time. Anyways, the decolorizer step pulls the crystal violet iodine complex out of the gram-negative cell, but not out of the gram-positive cells. So this is the differential step of the gram stain. And the cells are different at this point. The gram-negative will be clear. The gram-positive will be staining purple. You then apply the counter stain, which is safranin, and that will stain the gram-negative cells pink. But the gram-positive cells retain the crystal violet, so they will appear purple. Any question about any of that? All right, so then answer the question. Below are two smears viewed using the oil objective immersion lens that were stained with the gram stain. Complete the following table. The gram reaction, gram positive or negative E. coli, list that there. The gram reaction of the Staphylococcus aureus, list that there, gram negative or gram positive. And then give the cell morphology, the shape is the E. coli coccus or bacillus, is the Staphylococcus aureus, coccus or bacillus. And here's the picture, E. coli here, and Staphylococcus aureus. This is a better picture of the E. coli because the cells are not stuck all together, although there might be two stuck together right there and there, but most of them are individual bacilli. And then after that table, answer these questions. So this is procedure 3A. Staphylococcus aureus, after the primary stain is added, is what color? Pink, purple, or colorless? E. coli, after the primary stain is added, is what color? Pink, purple, or colorless? Staphylococcus aureus, after the alcohol acetone step, is what color? E. coli, after the alcohol acetone step, is what color? Staphylococcus aureus, after the counter stain is added, is what color? And E. coli, after the counter stain is added, is what color? So write the color there. Procedure 4, the acid-fast bacteria stain. We have a little video clip on performing the acid-fast stain. And this is the steps of the modified Kenyan acid-fast bacteria stain, or acid-fast positive stain. Now this B is not positive, it's bacteria. And that is the stain that you're viewing in the clip. Any question about any of that? So first you apply the carbol fusion. All the cells will stain pinkish from the carbol fusion. You then apply the acid alcohol wash. It'll be a decolorizer to pull the carbol fusion out of the acid-fast negative cells, but the acid-fast positive cells retain the carbol fusion. You then apply the brilliant green as a counter stain, and the acid-fast negative cells will stain green, and the acid-fast positive cells will stain pink. They're pink from the carbol fusion, and they're green from the brilliant green. Any question about any of that? All right, there's the steps for the acid-fast stain. and then answer the questions. See below are two smears viewed at 1,000 total magnification that were acid-fast bacteria stain, meaning we applied the acid-fast stain. Are the organisms noted by arrows acid-fast positive or acid-fast negative? Supply an answer for each arrow below. If you can, put your answer right by the arrow, but if you cannot, you can put it either here or here. Do something like in the first picture, the leftmost arrow is state what it is, acid-fast positive, acid-fast negative. And the second arrow is state what the arrow is. And then state on the second picture, the first arrow on the left. So this one you could say either the left or the right, or you could say the top or the bottom. On this one, say the arrow on the left and then the arrow on the right. State whether it's acid-fast positive or acid-fast negative. So you should have 4 answers for this picture. Okay. There is a way you could say something like all the cells that stain green or such. So you could possibly have 2 answers, but most people will have 4 answers. So you need answers to those arrows. Procedure 5, the special stains. View the following clips. We have a clip of making an endospore stain, and we have a clip for a negative stain to highlight the capsule. Below are 3 bacterial smears that are being observed at 1,000 times total magnification. And you have to sort of figure out what stain was used on 5-2A, 5B, and then 5C, and then answer the question. A goes to the picture A. This is the Schaefer-Fulton stain, which uses what as the primary stain and ultimately dyes what structure in the cell. And the structure is produced by some bacteria to aid in survival. Any question about that? So for A1, you're telling me what the stain is. And then for A2, you're telling me what structure is dyed by that stain. So that'd be A1, A2, then A3. What are some examples of pathogenic endospore producing bacteria? And you can get that answer, if you don't know, in the lab module. And then for 5B, B1, this is what stain, so name the stain, that coats long whip-like appendages which aid in motility. And then for 5B2, state the flagella arrangement type. Right there. And then 5B3, what are some examples of clinically significant bacteria that produce flagella? And that is discussed in the lab module. 5C, this is a stain which uses India ink or nigrosin and can be used to detect the presence of a cell structure. So list the stain in 5C1 and then list the structure stain in 5C2. And this structure is located exterior to the cell wall and prevents phagocytosis. And remember, C goes to picture C here. You can blow that picture up, by the way, so you can see that better. Yeah, that's better. Now let me shrink it down a little bit. And then 5C3, what are some pathogenic microorganisms that produce a capsule? And you can get that from the lab module. Those of you who do use AI to answer these questions, I will warn you that I'm only accepting answers that are discussed in the lab module. So you must give an AI answer that is in the lab module. So if you use AI to answer these questions, then make sure it's in the lab module. Any question about that? So if it isn't in the lab module and you use AI to give an answer, your answer is technically correct, but it'll be marked wrong because I'm only taking answers from the lab module. And in this one, I don't think it's going to be a hurdle for anyone, but in some future questions, it will be a hurdle, like these case studies. You can answer this case study just by putting it in AI, although I don't know how you put it in the pictures. But a 45-year-old female is experiencing some respiratory issues based on her clinical presentation. A doctor orders a chest x-ray and sputum for AFB, meaning acid-fast bacteria culture. In addition to the culture, an AFB smear is made and stained. Based on the findings below, what is the most probable diagnosis? And you must name the species causing the infection. So you must name the species by its scientific name, meaning give the genus and the species specific name, species specific epithet. Sorry. And there's the x-ray of the patient. You'll notice that this lung looks odd right there, kind of odd right there, and then odd right there. The lungs are normally all dark, like this part right there. And so this is something wrong with this patient in the x-rays. And we really should have a picture of a normal lung. But trust me, the normal lung looks kind of like all this section there. And that's odd. That's really odd right there. And then there's the sputum smear, and there's the Zeal-Nielsen stain made on the sputum. So give me the species causing the infection. And then case study 2, a 64-year-old diabetic patient is admitted to the hospital with soft tissue necrosis. There it is there, and that looks really bad. A routine culture and a gram stain is ordered. The bacteria grow aerobically. Based on the findings below, what is the most probable diagnosis? Once again, you must name the species which is causing the infection. You can't simply give me an answer like diabetic necrosis. Okay, you must name the species causing the infection. So there's the diabetic necrosis. There's the gram stain of the bacteria. You can see their rods, the gram-positive rods. And there's the Schaefer-Fulton stain. You can look up what that stain stains if you don't remember from the lab, and then list the species causing the infection. Any question about any of that? All right. That's it. I will be here until 4 o'clock to answer any questions, maybe 4:10, or until the last student leaves. So get started on the lab. If you have questions, I'll be here. But remember, ask your questions before the last student leaves, because once the last student leaves, I'm going to leave as well. On the lab.
Yeah, I have a question.
Uh-huh.
Between the shapes of the staphylococci and the regular cocci, is it just the fact that the staphylo one is just clustered?
It's asking for the shape of the cells. Is that what you're asking for?
No, just like in general, the shapes.
You're talking about the cellular growth arrangement?
Yeah.
All right. And what's your question again?
The difference between the cocci and the staphylococci?
Cocci is only the shape of the cell. It is not the growth arrangement. The staphylococci is a growth arrangement that grows where the cells are stuck together in a cluster.
Oh, okay.
Okay?
So then the cockeye is just like how the cell looks then.
Yes.
Okay, thank you.
All right.
So not all of them are shapes.
What?
So the diagram that we were giving on our lab, not all of them are shapes. Some of them are clusters.
There are three bacteria shapes, basically. Let me go to that. Three bacteria shapes. There's a cocci, there's a bacilli, and there's a spirilla. Those are the three basic shapes that bacteria have. There can be some other shapes like pleomorphic and coccobacilli, but most of them are either a cocci, a bacilli, or a spirilla, or some combination of these three. Like the coccobacilli is a combination of the cocci and the bacilli. So that is the basic shape of the bacteria. There's also a cellular growth arrangement, and that is how the cells stick together. You can have cells that do not stick together, and that would be a singlet, like a single cocci. There is also
Oh,
diplococci when that's where two cells stick together. And if you have multiple cells stick together in a row, it's called streptococci. You can have cocci stuck together as a tetrad. You can have cocci stuck together as eight cells. That's a sarxinia. And then you can have cocci stuck together in a cluster. That is a staphylococci.
okay, that makes way more sense. Thank you.
All right.